Today we are going to talk about phylogenetic signal. This is a really fascinating concept that I've explored quite often throughout my master's. We're going to break down the conceptual and theoretical components of interpretation. We are going to really focus on sort of the types of questions you can ask, how phylogenetic studies are conducted, and we're going to focus a little bit more on the interpretation of the results, right? Like, what does the detection of phylogenetic signal actually mean? So if you haven't been in one of these webinars before, you know, we don't go too in-depth into the statistics. We don't go into the equations as much. We're really going to focus on concepts in theory because you won't be able to understand the statistics unless you really understand that underlying theory. So first things first, let's just start with a really, really brief introduction to phylogenetic signal. So a traditional definition may be phylogenetic signal is the pattern by which related taxa resemble each other more than taxa pulled randomly from a phylogeny. So I'm using taxa here. Usually it's at the species level, but you can also run phylogenetic signal on populations, for example. And what we're saying here is that species or taxa that are more closely related tend to look more similar, right? In this little hypothetical example, we have hummingbirds down here on the left. They look more similar to each other than a hummingbird looks to, say, a hawk, right? Now, when we say the taxa pulled randomly from a phylogeny, that is essentially the null hypothesis, that there is no signal. And that looks like the sort of the graph on the left, right? So this would be a randomized phylogeny where all the tips are random in terms of their traits. They really have no inherent grouping. They are spread randomly throughout the phylogeny. And a tree with signal would be grouped according to their relatedness. Now, we should note here that this is kind of the conceptual, the theory, the getting into the statistics of it, right? We're talking about random trees. Well, in nature, we don't often have fully random trees, but this is what they are talking about when they say a random tree, right? So this is just cementing the ideas. And when we say traits, that really can be anything. There's a wide variety of traits that you can use, as we will see in the case study examples at the end of this presentation. So you can use geographic, physiological, life history, behavior traits. There's a wide variety of eco-evolutionary traits that you can use. It really depends on your research question, on what data is available, and so many more considerations. So from here, just know whenever we're saying traits, this could refer to any of these traits. So let's give a very good example for what strong signal looks like. So this is strong signal or high signal in the literature, and maybe this would be a perfect example of just strong signal. We're using a very simplified explanation. So this is a phylogeny. Of course, if you're struggling with how to read phylogenies, I actually have a whole video on that. If you're watching the recap, I'll link it. But just know that we can, I do have a video about phylogenies and how to read them. So this is strong signal. And we know it's strong because we can see that the shapes are clustered in ordered of their relatedness. So the triangle up here is in this clade that has all of these triangles that are really closely related to one another. The circle is, in this circle clade. The square is in the square clade, right? So these are, in this case, perfect monophyletic groups. But the essential theory that you really need to understand from this is that, well, they are more closely related, ergo they look more similar. Versus something that would be weak signal. If it were talking about the shapes here, this is what it would look like, right? The triangles and the circles and the squares, they're distributed throughout the phylogeny with no inherent grouping. So this would be no signal or weak signal. But remember, we can look for signal across many different traits. And in fact, if you incorporate color into this hypothetical example, we actually see that while there is very, very weak signal for shape, color actually has very strong signal, okay? And this is something that I really want to emphasize, is that signal isn't just owed to the entire species, right? It is trait dependent. And in fact, one of the case studies we'll look at looks at this particular concept quite in depth. So, in essence, context matters, particularly when you're answering your questions. Now, of course, a phylogenetic signal is just a pattern, okay? Usually, we are interested in the processes behind the pattern, right? We detect signal or we detect no signal, and we want to understand why. So let's start with high or strong signal. There's a wide variety of different traits that can affect it, a wide variety of different processes that can affect that pattern. So we're just going to focus on a few here. One of them can be very strong selection. Imagine populations of frogs, species of frogs, where for some reason there is high sexual selection for red eyes, right? And as you can see, this little blue-eyed frog down here is alone. There is a strong selective pressure. This will lead to high signal in the phylogeny, okay, for red eyes. So this is one great example. Usually whenever we see high signal, there could be some strong selective pressure. In this case, it's sexual selection, but it could be really anything. If you imagine a homeobox genes, right? Something that is very critical for the development of organisms, that is very likely going to exhibit very high signal. And sort of with this, also high gene flow, okay? So if we have multiple species, multiple populations, and they are consistently and with a large degree exchanging genetic information, they are going to become more similar over time, right? They are more closely related and they are more similar. They're swapping genes. So this is going to create high signal, even if there are individuals or species that are different, such as this little blue-eyed frog. Population genetic theory would say they would likely be removed from the population over given enough generations and if drift just so happens to do that. So this is a classic example, more focused on population level rather than species level. But over time, niche conservatism, right? This concept that a organism has a niche and it doesn't really change over evolutionary time. This can result in high signal. So what you'll see is a general trend in the literature to refer to high signal traits as those that are phylogenetically conserved. They are not that variable, okay? So this is an example of high signal, and again, there are many, many processes that can create this pattern. So, of course, what about low signal? In this case, again, the lowest of low signals. If there's absolutely no signal in the phylogeny, that means that the trait is effectively random. So these are traits that are going to be assumed to be more adaptive, a little bit more labile, or they have some evolutionary process that creates this signal. One of which is an adaptive radiation, okay? So in this example, there was a common ancestor, the green frogs. They diversified into a diverse array of habitats, into the forests and the streams and the mountains and the desert, and through there, their color changed over time. If you plotted these frogs on a phylogeny, they would be closely related. It's a recent radiation. It's a recent speciation event, right? But those traits are then not consistent. They are not all green frogs with red eyes. They may be the red frog with the green eyes or the brown frog with the blue eyes, etc., etc. So this is an example of what would create low signal. Another one is actually convergent evolution, okay? And you can see here, if we were looking at the skin color of frogs, and that is the trait that we are trying to detect signal on, we had some mutation that caused green frogs early on in the phylogeny and another mutation that caused orange frogs way back in the phylogeny, right? Their common ancestor may have been some other frog, right? A blue frog, for example. But then, along that orange clade, another mutation occurred for green frogs. And now it's a convergent evolution event. If this event didn't happen, we would likely see high signal, right? One clade is all green frogs, one clade is all orange frogs. But now the results are a little bit more confusing. So convergent evolution can absolutely create low signal. But also, divergent evolution can create low signal. Interestingly enough, divergent evolution can create low signal depending on the length of the branches, right? So say this, in this example here, this was a very recent divergent, say a river came through 20,000 years ago, and now we have these two populations that are highly diverged based on color. In the context of a larger phylogeny, this may be very, very low signal, and it's only because the trait is more spread out, right? So both convergent and divergent evolution can create low signal. The difference is context. The difference is how are these traits spread across the entire phylogeny? In this case, we're only using 5 frogs. You would hopefully use many, many more species, many more populations. So just be very, very aware of a few things with signal. You'll also notice that in the original slide where I'm showing low signal and high signal, I mentioned evolutionary rate. I will say that in the literature, there is a decent amount of argument on whether evolutionary rate, the rate by which a species evolves, is actually detectable or has an influence on signal. It does go back and forth. I would say exercise caution on your results, and especially exercise caution when interpreting results of this. And remember that signal is just a pattern, okay? It is not the process. Signal isn't creating some new species. Signal is being created by processes. And that same signal can be caused by many different processes. You can't just necessarily assume a process just by the detection or not the detection of signal. And kind of in that same vein, weak signal is just as interesting as strong signal. In this case, I'm showing off, African cichlids, one of the classic examples of niche specialization, adaptive radiations. And in this case, they would have absurdly weak signal for a trait such as color or pattern or life history. And that's because of that radiation. Now, so this is this kind of trying to break down this idea that you need to have significant positive results in order for your research to be interesting. In this case, the absence of signal is a really cool phenomenon to explore, right? Now, I want to move on in this presentation about how to actually detect phylogenetic signal and interpret results. Now, of course, with methods, I do keep these very, very brief. I encourage you to actually look into your own studies because, to be quite honest, going through the methods of how it is detected will take a whole lesson, probably multiple. But in general, the null hypothesis is that there's a random phylogeny, and then compared to this random phylogeny, we can give it our actual phylogeny, right? So this is a permutation test. This is what the very early methods of signal used. And they really just look for high signal and low signal, and they can quantify it. There are a lot of different methods. Some of them were like Moran's eye, which was from spatial autocorrelation, is also used. If you've RSVP'd, there will be a recap email. If not, go to the recap page. That'll be linked at the bottom of this video. There I will have further sources for how to actually measure and test phylogenetic signal that covers it in much more detail than I can in this presentation. But we are going to focus on two methods that are widely used in the literature, and they are often used together. These are both maximum likelihood methods. They do utilize the permutations at a certain component to actually test if the differences are significant according to a null model. But we're really going to look at Pagel's lambda and Blumberg's k. So these are very, very simple to interpret. Both of them follow a very simple metric where 0 is no signal and 1 indicates very strong signal. The actual methods for how they do this differ a little bit. Again, go into the other literature to get these nitty-gritty specifics. But any value in between 0 and 1 can be considered weak or semi-strong signal. I've seen authors use a value of 0.5 as their cutoff for moderate strong correlation. So anything below that would be considered a weaker signal, and then anything above 0.5 may be a strong signal. And similarly, Blumberg's K is the same. 0 is no signal, 1 is strong signal. You can have values theoretically over 1, and these use a Brownian motion model of evolution. Won't get too in-depth into that. But here, they're just saying that there is more signal than you'd expect at random. So there is some debate on if this is actually that relevant or if this is more of a statistical artifact, but either way, they are detecting signal. And they are interpreted in our results. So, like I said, I really want to focus on the theory, concepts, and interpretations. So, we're going to look at the interpretations of P equals lambda, Blomberg's K, in 3 different examples. These are 3 studies that will be linked in the description down below and on the recap page. And example 1 is probably the best paper to read if you want to understand signal. And this one is Phylogenetic Signal in Primate Behavior, Ecology, and Life History from Jason M. Kamilar and Natalie Cooper. So this one is exploring signal across traits in primates. And the reason they did this study is because there are a lot of assumptions of signal. This paper is half a review paper and one that just gives a lot of clarity into how signal actually operates. Because for a long time, some traits were assumed to have low and high signal. But they show that's not always the case. So what they did is they went through the literature and they found 31 variables for 213 primate species. They incorporated a phylogeny and they broke those variables down into 9 trait categories. Those are body mass, brain size, life history, sexual selection, social organization, diet, activity budget, ranging patterns, climate, niche space. So many more variables were found. I think I misspoke earlier. These are not the 9 trait categories. These are just some of the variables they have. Or they were. We'll find out. So, what are the results? They found that nearly all of the traits exhibited significant signal. So almost everything had some degree of signal. All of them were significantly different from 0. But only 8 of them had a moderate to strong signal. So these are traits like mean brain size, relative brain size, mass of the individuals, gestation length, okay? So only 8 of the 31 variables actually exhibited very strong signal. In fact, most of them were much lower. And this indicates that many traits are actually variable along evolutionary time. So traits like maximum longevity only had a signal value of 0.25. This is very weak signal. So this may assume that longevity can actually vary within and across species over time. That's sort of what this low signal is saying. They also found that brain size had a K higher than 1. Again, it was more conserved than we'd expect from random, right? It was very highly conserved. So this is saying that there might be a very strong selective pressure for large brain sizes in primates, which is kind of in line with a lot of our theory. But one of the best findings is that those values ranged massively within groups. Groups like life history, which includes gestation length, litter size, interbirth interval, weaning age, maximum longevity, they ranged from 0.705 to 0.263. They had weak signal and strong signal. And also things like ranging patterns, right? So these would be like territoriality, home range, day range. They also exhibited strong and weak signal. So really, you just can't assume that a signal will have, or a trait will have some signal just based on larger categories. You cannot make that assumption. But I want to move on to example number 2. So again, if you really want to examine the information and really try to understand signal, I'd highly recommend this paper. It just does such a phenomenal overview. This will be linked as well. Now, example number 2 is a great use case, okay? What they're doing is they're testing for signal in the flowering onset times for 116 families of flowering plants. Why would they do this? Well, we know that climate change affects flowering times, which could be a dramatic conservation impact for flowering species, right? The thought process being if they flower at the wrong time and their pollinators are not active, then the plant will not get pollinated, right? So we can actually use signal to detect groups that may be at greater risk. The assumption being that high signal groups of plants that have very strong signal, will not be as adaptive to change, so they may be at greater risk for climate change. So, how do we do this? What they did, what the authors did, is they did a literature review for data sets where it shows flowering onset through time. Some of these data sets spanned multiple decades. So, from that, they could say species A has over time delayed their flowering, or they haven't changed their flowering, or they're actually advancing their flowering onset and flowering earlier. So we can see, hey, these plant species are changing their flowering onset. Can we detect if certain groups are more adaptive to flowering onset? Can they have a little more wiggle room about when they flower? What they actually found was really fascinating. So they did detect signal in nearly all the groups. Okay, this phylogeny right here is just showing the shift in flowering onset. Red means it's earlier. Blue means it's later. What they did find, even though signal was found in almost all the groups, it was very low. They actually indicates that flowering time is rather flexible. So these groups with this low signal may be more resilient to climate shifts. It's possible that the traits are a little bit more labile. Of course, this was needs more research, needs more done. Flowering onset is not the only trait that may be affecting species, but if we're truly worried about flowering onset, then maybe we only need to look at the groups that actually did have that high signal. Maybe we can feel a little bit better knowing that groups with low signal are probably adapting to change a little bit easier. So this is a really great conservation use case for phylogenetic signal. And the last one is actually the paper that kind of inspired me to make this because I thought it was such an amazing use of signal. What they're doing in this paper, Behavioral Plasticity and the Origins of Novelty, they are testing the evolution of the rattlesnake's rattle. How did the rattlesnake even get its rattle? And the reason they're doing this is because studying the evolution of complex traits is a lofty goal in evolutionary biology. But it's really hard. It's really hard to study how something that likely iterated over many, many steps, hundreds and thousands of steps, can actually come to fruition, right? In this case, they are able to use signal, among other methods, to test how behavior may actually lead to novel morphologies like the rattlesnake rattle. How did they do this? Well, they measured the tail vibration rate and duration for 56 species, and not just rattlesnakes. They measured this for other species, even non-venomous snakes, okay? Many snakes will rattle their tails as a predator deterrence, or in some cases to actually attract a predator towards eating the tail instead of eating their head, right? So tail vibration is found throughout snakes. It's fairly widespread trait. But they were measuring the vibration rate and duration. They were then able to incorporate phylogenetic information and do ancestral state reconstruction. But for the purpose of this presentation, they really focused on phylogenetic signal and the relatedness of the organisms. So what did they find? They found phylogenetic signal was very strong for rate and duration, indicating there was possibly strong selection for rattling behavior or tail shaking behavior. And by incorporating that genetic relatedness, they were able to see that snakes which are more closely related to rattlesnakes exhibited a more rapid rate and a longer duration of rattling. Which, as you can see from the figure here, the rattlesnakes, being this green and the only clade with a red, they have very high rates and very high durations of rattling. And they were able to use phylogenetic signal to help answer this question. Again, I really like to see phylogenetic signal as just a tool in the toolbox of biologists. So, this presentation covered phylogenetic signal. This is actually right at the end. We try to keep these 20-30 minutes long. We will have time for questions and answers. Just know, I will be putting up a recap as well as the handouts for this webinar will be up on the website pretty soon, actually. So these handouts are just very quick little guides for phylogenetic signal, as well as incorporating some extra sources for you to learn more. Next week, we will be doing a very straightforward webinar about p-values and really trying to focus in on the difference between significance and meaningful data, right? Meaningful results. So that'll be next week, same time, right here. There will be an email to RSVP. But for now, are there any questions? Anything at all? You can just leave it in the chat. Phylogenetic signal is a really fascinating concept that I've been having more and more ideas about how to explore for my own stuff. It's incredible, really. Like the more you think about it, it's so cool. Yeah, of course, Jenny. I love doing these presentations, so yeah. Oh. And yeah, we are focusing on these handouts to be very, very simple, and they're free to download. We accept optional contributions, but they're 100% free. Am I aware of any literature on phylogenetic signal in fungi? That is a very good question. Off the top of my head, no, and I think that's more of a indicator of my like taxonomic study choice. But let's actually see really quick. Let's just do it. I'm almost certain there would be signal, and then there was a very recent yeah. So a lot of times, here's a good example here. Kingdom-wide analysis of fungal protein coding in tRNA genes. Let's make this a little bit larger for y'all. Reveals conserved patterns of adaptive evolution. So yeah, so there absolutely is literature out there for the fungi world. And this is a cool example too, that you can actually look at the individual protein coding genes and detect signal. So I'm just going to go in order of these questions. Can you walk me through the adaptive radiation bit again? Absolutely. So let's go here. Yeah, because adaptive radiation one, I do notice that I think high signal is a lot more intuitive than low signal. So let's do adaptive radiation. Let's get to this example here. So remember that low signal would indicate that a trait is more variable, right? It indicates that a trait can be randomly spread out. So in an adaptive radiation, this would assume that, they have some common ancestor. In this case, it's this green frog. And then they radiated outwards. Maybe they went to different habitats, or maybe they are the African cichlids, right? Where it is microhabitats, little areas within the lake. But regardless, they have diversified, okay? They have diversified in some way. And remember, in this case, in signal, it depends on which trait you're looking at. If we're looking at body morphology of these frogs, they would exhibit extraordinarily high signal, because in this example, they all have the same body morphology. But if we're looking at the color of the frogs, we would likely see very low signal. And that's because that color is so different, okay? There are 5 different states here. We have green frogs, red frogs, brown frogs, orange frogs, blue frogs, right? So in this case, that signal, there really is no signal, right? If it were the case that this has now been 20 million years, and now these frogs have all evolved into different individual clades that have speciated dramatically, and now there's 30 red frogs and 20 blue frogs, right? Then we may detect high signal, but usually in the context of adaptive radiation, it's a recent radiation. So that's what would make it low signal. I'm hoping that helps cement it down. And since we also kind of touched on the next question about how do we determine sampling size, should we always include as many species as possible? There is some discussion about this in the literature. I believe actually that Primate paper that I linked mentions it. In general, if you can add more species, do it. Keeping your research question in mind, but you have to think about what will happen if you remove species. You could very, very easily. Let's actually show an example, right? Let's say we're doing this divergent evolution, okay? And um, let's say this is what we're going to study. Like this is our study system, okay? And we decide, oh, well, we don't have the material for these two green frogs, so we're going to exclude them from the study. Or we don't want to sample them, they're in a different genus, right? Well, what happens if you exclude these two green frogs? Oops, apologies. If you exclude these two green frogs, you're only going to be looking at the orange frogs. So that's actually going to give you an example of high signal, right? Because you don't have that variation. So in general, sample size, keep it fairly large. I think in the literature, the number I have seen is at minimum 20. But it depends a lot on your study system, on which metric you're actually using. So that's where that metric paper the methods paper that kind of goes a little bit more in depth as to which ones. But yeah, I mean, if I guess the answer is why not include more in a good way, um, other than time and cost of doing all that, right? Yeah, the macroscopic features are so ambiguous with so many fungi. Well, yeah, but also like it doesn't have to be the macroscopic features, right? I mean, yeah, we can do the color, the number of gills, or the presence or absence of gills, right? Um, and if I say anything weird about fungi, it's because I don't know as much as I should. But it could literally be the geographic range of fungi, right? Which would be a weird one of doing, but it'd be interesting. It could be, I mean, any number of things. It could be when they actually like what conditions cause them to sprout from the earth. Are so like there's a lot of different ways to answer this question, right? Like remember, if we go to the traits, boom, like there's so many different traits you can utilize for signal papers. And so, do some papers assume the limited sampling is representative of the clades, e.g., for the plant paper? I'm sure they don't have data for every species. Yeah, so I mean, at some level, you always have to, right? Um, I work a lot. Like my current research incorporates species that don't have any genetic data onto the phylogeny. That's a whole other methodology at some point. Um, they assume that limited sampling is an issue. I think that it's not as big of an issue nowadays. Most papers that are published recently, there's almost always a global time calibrated phylogeny for most of the organisms, right? Um, but yeah, anything from like 10, 20 years ago, they may be missing a lot of species. And sometimes it is also that the trait data is missing. Again, that's getting a lot better. So this is sort of a maybe a bigger issue that is in issue in like most phylogenetics is that limited sampling does affect it. They usually mention it in the paper if it's of a huge concern. Um, but then we can get into the issues of, well, they don't account for extinction in their results, which extinction would have a massive bias on our results as well, for any phylogenetic study. Um, I think my favorite example, which is one I'm going to save for island biogeography, but yeah, okay, yeah. I mean, missing trait data, same thing. I mean, there's ways to estimate it, of course, but yeah, no, it's tricky. It's tricky, but I mean, same thing. It's still species get removed from the analysis for um, not bad reasons, but just sampling reasons. So they have to assume it. If you can only get like one or two representatives of all your clades, maybe rethink your study. Use different trait data. But some people do this in a matrix sense where they'll have the 30 some odd traits and the 200 species, but then for each you analyze each trait individually. So you can do just brain size, even though you're missing it for 20 species, and then run another signal analysis on mating structure, because that one's only missing for 5 species. So you can run it down the list and just be very aware of any implications that has. I'm sure there's a way to estimate or approximate, maybe that's a better way of putting it, approximating the missing trait data. Um, but that gets a little more tricky, right? And it, I don't know, what does it really add to the analysis? Right. Okay. Well, I am not seeing any more questions, and thank y'all for showing up. So, like I said, the recap will be posted. I usually get those up by Friday. That'll include the full video with like chapter markers so you can bounce around, but also these handouts I'm working on. Those will be done and uploaded. You can download them for free. They're just little quick summary guides of this presentation, or like cheat sheets. And yeah, next week is about p-values. If there's ever a webinar that you wanted to, if you want to see happen, any particular individual topic, anything, feel free to comment it down below, shoot me a DM, anything like that, because there are so many topics we can go down into. And this was actually our first conceptual topic, and I think we're going to do way more of them because they are way fun. But cool. Thank you all for showing up. I love doing these streams, and yeah, see you at the next one.